Hello everybody, my name is Amir and this is Learnoplasty, in which we make your previous learning beautiful and understandable with a surgery. This video is about peptide bond and peptide group. In the late 1930s, Linus Pauling and Robert Curry, with the help of X-ray, started a series of studies that laid the foundation for our current understandings of protein structure. So Linus Pauling and Robert Curry realized that this peptide bond is different when comparing to other single bonds. For example, if we compare the length of this peptide bond with the adjacent nitrogen to alpha carbon single bond, we see that this peptide bond is 0.13 angstrom shorter. Or we can also notice that this peptide bond has less freedom for rotation. As we know, groups around a single bond can rotate freely and have different conformations, but that is not the case with this peptide bond. They also found out that this carbon-oxygen double bond is 0.02 angstrom longer than other carbonyl groups that we see in, like, for example, aldehydes or ketones. So they came up with this idea that because of these resonance interactions, give this peptide bond 40% double bond character. So that's why it's a bit shorter and has less rotations. Another interesting fact about peptide bond is that the peptide group is almost always in trans conformation. And we can show this in two ways. Number one, two successive alpha carbon atoms are on the opposite side of the peptide bond. And number two, the oxygen of a carbonyl is on the opposite side of the hydrogen of the amide. So, and the reason with that, it's because in this way the steric interferences is the least. And by steric interferences, I mean when two non-covalent neighbors are closer than their van der Waals radius. For example, this is a cis conformation for two peptides, for one single peptide bond and two amino acids. And as you can see, the two successive alpha carbon atoms are both at one side of the single peptide bond, or the oxygen of a carbonyl is on the same side as the hydrogen of the amide. So in this case, in cis conformation, you can see the two R groups have some steric interferences, which is not thermodynamically favorable. Although the peptide bond doesn't rotate freely because of this 40% double bond character, but the other two adjacent single bonds in the backbone of a peptide can rotate, and we can acknowledge the position of the peptide groups by knowing the angle of these two bonds, which are called torsion angles. So here I drew two amino acids and one peptide bond, and I put some numbers so that I can explain more easily. So here we need your imagination so that you can understand these angles. Imagine you are this nitrogen number 1 and you're looking to this alpha carbon or carbon number 2. This would be the view. This would be the view. So this blue circle is you, and you're watching the black circle in front of you, which is the carbon number 2, and the rest are the bonding groups that are attached to either the nitrogen, or the carbon you're looking to. So this angle between the two carbonyl, one carbonyl and two carbon, between these two carbonyl, this angle is called phi angle. We can also say the angle between carbon, nitrogen, alpha carbon and carbon. And if you're the alpha carbon, or of the carbon number 2, and you're looking through the carbon number 3, which would give you this view, then the angle between the two amide groups, which be like this, would give you the angle called psi, or you can also say the angle between nitrogen, alpha carbon, carbon, and nitrogen. It is important to know that when the fourth atom, for example, here this nitrogen rotates clockwise, for example, this, if this nitrogen rotates clockwise around this first nitrogen, like the first atom and the fourth atom. So the fourth atom rotates clockwise around the first atom. In this case, the psi angle increases, and it would be the same for phi angle too. But can you tell me in the comments what would be the first and fourth atom in this case? So we said earlier that groups in a single bond can rotate freely and create different conformations. But the thing is that in a polypeptide with different amino acids with different R groups, you cannot rotate in every angle because there might be steric interferences which is not thermodynamically favored. And that would answer the question, how can a polypeptide fold as folding decreases entropy which is not thermodynamically favored? Well, the answer is if you could rotate in every angle, I mean, if any phi or psi angle could be possible, then there would be so many different conformations for a polypeptide, and this huge increase in entropy would never let the polypeptide to fold, right? But in reality, not every phi and psi angle is possible due to steric interferences. So this rigidity of the peptide unit and the restricted set of allowed phi and psi angles limits the number of accessible unfolded conformations sufficiently to allow protein folding to take place. Ramachandran recognized the allowed values for phi and psi angles in different secondary structures that exist in nature, for example, alpha helix or beta sheet. So he visualized them on Ramachandran plot. As you can see in this plot, there are 3 regions that are sterically allowed phi and psi angles. These regions are actually the conformations that usually exist in nature, such as alpha helix or beta sheet, which we discuss about later. And here are some questions to double check your learnings. Thanks for watching. Don't forget to subscribe to this channel.